This is chapter 9. We're going to start discussing muscle tissue. So in this chapter, we're going to look at the structure and function of the 3 different types of muscular tissue. We're going to look at what happens at the neuromuscular junction. We're also going to look at how muscle cells use energy and also how muscle tension is controlled. Now there are 3 types of muscular tissue. These are skeletal, cardiac, and smooth muscle. And even though they share many properties, they do differ from each other a lot in their microscopic anatomy, their location, and also how they're controlled by the nervous and endocrine systems. Now, muscular tissue as a whole has 4 key functions. Our first one is going to be producing body movements. You know this because we know that we move our limbs by muscles. Our second function is going to be stabilizing body positions. You can think of your skeletal muscles helping you sit up straight in your chair or stand on one leg, for example. Our third key function of muscular tissue is going to be storing and moving substances. For this one, you can think of how smooth muscle helps to move food that you eat along your digestive tract. We will be talking about these in more detail later, but just a quick example. And then our last key function of muscular tissue as a whole is going to be generating heat. So you can think of shivering is a quick muscular contraction that helps to generate heat in your body. Now, skeletal muscle tissue is named because most skeletal muscle moves bones of the skeleton. There are a couple skeletal muscles that attach to and move the skin, which are going to be muscles of facial expression, but most skeletal muscle moves bones. Now, skeletal muscle tissue is striated. Now, this is a term that means alternating light and dark protein bands. I can write this in here. Now, we'll see those alternating bands a little bit later, but these would be called striations. Now, these bands are seen when tissue is examined with a microscope. Skeletal muscle is also multinucleated, so there's multiple nuclei per muscle cell. And skeletal muscle works mainly in a voluntary manner, controlled by your somatic system or your voluntary system, which means that you consciously make the choice to contract and move your skeletal muscles. Next, we have cardiac muscle, and only the heart contains cardiac muscle tissue, which forms most of the heart wall. Cardiac muscle is also striated, but it is involuntary. The alternating contraction and relaxation of the heart is not consciously controlled. Rather, the heart's going to beat because it has a natural pacemaker that initiates each contraction. This is really a built-in rhythm that we call autorhythmicity. Sorry, that's... Gotta spell this right. So it's autorhythmic. so it's not consciously controlled. It has this automatic rhythm to it. There's going to be several hormones and neurotransmitters that can adjust heart rate by either speeding it up or slowing down the pacemaker. Then we have smooth muscle tissue. This is sometimes also called visceral muscle tissue. And it's going to be located in the walls of hollow internal structures, things like blood vessels, airways, and most organs in the abdominal pelvic cavity. They are also found in the integument attached to hair follicles too. Now, these tissues lack the striations that we see in skeletal and cardiac muscle, so they're going to be non-striated, so therefore smooth. That's where they get their kind of smooth muscle term from. Now, the action of smooth muscle is usually involuntary, and in some smooth muscle tissue, such as the muscles that propel food through your gastrointestinal tract, also have some autorhythmicity. Both cardiac and smooth muscle are regulated by neurons that are part of the autonomic division of the nervous system. And they're also controlled by hormones released by endocrine glands. So we can put this over here. Regulated by the autonomic nervous system and the endocrine system hormones. So here is just a really quick visual representation of our three types of muscular tissue. The top one we have is skeletal muscle. We can see each of these cells. So one of these is going to be one cell with multiple nuclei per cell. We can also see these bands here. These are going to be the striations. You can see these bands in the cell. So that's going to be skeletal muscle. Smooth muscle, you can see that these are going to be really smooth muscle cells, no striations, single nuclei. Cardiac muscle, you could still see those striations. There aren't any nuclei present or visualized in this picture, but they do have single nuclei, and they also have these interconnections, which we will talk about a little bit later. Now, there are four main properties of muscular tissue. The first one being electrical excitability. So this is a property that means that cells can produce an electrical signal, which is called an action potential. So an action potential is just an electrical signal produced by cells. Now in muscles, these are called muscle action potentials. And nerve cells can also produce action potentials, but in nerve cells we just call this a nerve action potential. Our second property is going to be contractibility. This is the ability of muscle tissues to contract forcefully when stimulated by an action potential. When skeletal muscle contracts, it generates tension, which is a force of contraction, while pulling on its attachment points. So these are going to be pulling on the skeletal muscle or skeletal bones. Now in some muscle contractions, the muscle develops tension, but it does not shorten. Next, we have extensibility. So this is the ability of muscular tissue to stretch within its limits without being damaged. Now, the connective tissue within muscle limits the range of extensibility, and it keeps it within the contractile range of the muscle cells so that it doesn't get damaged. Now, normally, smooth muscle is subjected to the greatest amount of stretching. And then we have elasticity, which is the final property of muscular tissue. This is the ability of muscular tissue to return to its original length and shape after contraction or extension. Now for the rest of this chapter, skeletal muscle will mainly be the focus, even though we are going to mention smooth and cardiac muscle briefly. So we're going to go into a bit more detail about skeletal muscle. So each of your skeletal muscles is a separate organ composed of hundreds to thousands of cells, which are called muscle fibers. And they're called muscle fibers because they have really elongated shapes. Muscle cell and muscle fiber are going to be two names that mean the same thing. Two names for the same thing. Skeletal muscle also contains connective tissues surrounding muscle fibers and also surrounding whole muscles, as well as blood vessels and nerves. The subcutaneous layer, which separates muscle from skin, is composed of areolar connective tissue and adipose tissue. The term fascia, this is a dense sheet or broad band of irregular connective tissue that lines the body wall and limbs and supports and surrounds muscles and other organs of the body. Fascia allows free movement of muscle, carries nerves, blood vessels, and lymphatic vessels, and also fills space between muscles. So in this picture on the right here, we can see an entire muscle fiber, which is what this circle is around. So this is one muscle fiber or a muscle cell. And muscle cells will combine to each other to create a fascicle, and then multiple fascicles will combine to create the entire skeletal muscle. Now there are 3 layers of connective tissue that extend from the fascia to protect and strengthen skeletal muscle. So first we have the connective tissue surrounding the entire skeletal muscle, which is the epimysium. So this is, we can kind of label this as like the first layer of connective tissue. This is going to be the outer layer encircling the entire muscle, and it consists of dense irregular connective tissue. Next, we have perimysium. This is a deeper layer of connective tissue. You can call this the second layer of connective tissue. It's also a layer of dense irregular connective tissue, but it surrounds groups of 10 to 100 or more muscle fibers, and it's actually the perimysium that separates these muscle fibers into bundles, which are called fascicles. Now, many fascicles are large enough to be seen with the naked eye. And then we have endomysium. This is going to be the innermost layer of connective tissue, and it penetrates the interior of each fascicle and separates individual muscle fibers from one another. So it surrounds each individual muscle fiber. Now, all 3 of these layers are continuous with the connective tissue that attaches skeletal muscle to other structures like bone or tissue. Bone or other tissues like another muscle. Now they can extend beyond the muscle fibers to form a rope-like tendon that attaches the muscle to the periosteum of the bone. So a tendon is going to be continuous with the epimysium and the perimysium and the endomysium. Now muscle is really cool because it can regenerate itself. Now muscle stem cells, called satellite cells, These divide during injury or during development to produce many myoblasts. These myoblasts can then fuse together to form a multinucleated muscle fiber. So you can see they kind of fuse together to produce this immature muscle fiber. You can see these multiple nuclei within that muscle fiber cell, and this is from the fusion of all of those myoblasts. Now there's always going to be satellite cells around muscle fibers that can divide when needed to provide more muscle tissue. Now let's look a little bit closer at the microscopic anatomy of muscle. Now the multiple nuclei within a skeletal muscle fiber are located just below the sarcolemma. Sarcolemma, this is going to be a special name for the plasma membrane of a muscle cell. So plasma membrane of muscle cells is called the sarcolemma. So you can also see the sarcolemma on this picture too on the right, a little bit more zoomed in here, a little bit more details here. Now there are thousands of tiny invaginations of the sarcolemma, which are called transverse tubules. So they're going to be these little blue invaginations like here and here. These are invaginations of the sarcolemma. And these tunnel in from the surface toward the center of each muscle fiber. Now, because T-tubules are open to the outside of the fiber, they're going to be filled with interstitial fluid. Muscle action potentials travel along the sarcolemma and down through the T-tubules. So an action potential, abbreviated AP, is going to travel along the sarcolemma and down through the T-tubules, quickly spreading this action potential throughout the muscle fiber. So we'll kind of look at a little bit more detail at what happens when an action potential reaches a muscle fiber and how that action potential travels throughout the entire muscle fiber very quickly because of these transverse tubules. We'll look at that a little bit later. Now within the sarcolemma is the sarcoplasm. And now sarcoplasm, again, is just another special name given to muscle fiber cells, but this is just the cytoplasm of a muscle fiber. Sarcoplasm includes a substantial amount of glycogen, which is a large molecule composed of many glucose molecules, and glycogen can be used for the synthesis of ATP. So glycogen can be used to make ATP. In addition, the sarcoplasm contains this red-colored protein, which is called myoglobin. So this is a red-colored protein within the sarcoplasm, and this protein is only found in muscle, and it binds oxygen molecules that diffuse into muscle fibers from the interstitial fluid. So myoglobin is an oxygen transport molecule. Myoglobin is going to release oxygen when it's needed by the mitochondria for ATP production. Now in this microscopic view, we can also see a lot of mitochondria. Give me these little orange bean-like structures all around. Now, the mitochondria, they will lie in rows throughout the muscle fiber, and they're going to be found strategically close to the contractile muscle proteins that use ATP during contraction, so that ATP can be produced as quickly as it's needed. Now, these are going to be the muscle or the contractile muscle proteins. Here we have a sarcomere. The sarcomere are going to be the contractile units within the muscle fiber. So this is going to be the smallest unit that contracts within the muscle fiber. And here we have these little contractile muscle proteins, myosin and actin, within the sarcomere. So here we have really high magnification. We can see the sarcoplasm here is going to appear to be stuffed with little threads, these little threads here. Now these little structures are called myofibrils, labeled here. Now these are going to be the contractile organelles of skeletal muscle. Myofibrils actually extend the entire length of a muscle fiber, and they have these really prominent striations that make the entire skeletal muscle fiber appear striped or striated. A fluid-filled system of membranous sacs, which are called the sarcoplasmic reticulum here. These encircle each myofibril. So this picture is really kind of just a representation. It's very simplified, but these sarcoplasmic reticulums will encircle the myofibrils. It's a really elaborate system, but these SRs are very similar to the smooth endoplasmic reticulum in non-muscular cells. They have dilated end sacs in the sarcoplasmic reticulum, and these are called terminal cisterns. And these will butt against the T-tubule from either side. And this transverse or T-tubule and the two terminal cisterns on either side of it form what's called a triad. In a relaxed muscle fiber, the sarcoplasmic reticulum stores calcium ions. So as we can see from this key down here, the calcium ions are going to be these little purple circles. We can see the sarcoplasmic reticulum is full of them. So this is in a relaxed muscle fiber. Calcium is stored within the sarcoplasmic reticulums. Release of calcium from the terminal cisterns of the SR, this is going to be one of the factors that triggers a muscle contraction. Within myofibrils are smaller protein structures which are called myofilaments. Sometimes these are just shortened to filaments. But myofilaments means muscle filaments, so they're called myofilaments. The two types of myofilaments are the thin filaments, or actin, and thick filaments, which are myosin. Thin filaments are about 8 nanometers in diameter. They're composed of the protein actin, while thick filaments are about double the size and diameter, and they're composed mainly of the protein myosin. Both thick and thin filaments are directly involved in the contraction process. Overall, there's going to be 2 thin filaments for every thick filament in the regions of filament overlap. So you can see that here. We have 2 thin filaments and 1 thick filament where those filaments overlap. Now the filaments inside a myofibril do not extend the entire length of the muscle. Instead, they're going to be arranged in compartments, which are called sarcomeres. And sarcomeres are going to be the basic functional units of a myofibril. Sarcomeres extend from one Z disc to the next, and then this is where a new sarcomere will begin. The narrow plate-shaped regions of dense protein material, these are called the Z discs. You can see them here. And these are going to separate one sarcomere from the next. So a sarcomere is going to extend from one Z disc to the next Z disc. Now the extent of overlap of the thick and thin filaments depends on whether the muscle is contracted, relaxed, or stretched. Now there's the darker middle part of the sarcomere, which is called the A band. This extends the entire length of the thick filaments. Towards each end of the A band is going to be this zone of overlap where the thick and thin filaments lie side by side. The I band, this is going to be a lighter, less dense area that contains the rest of the thin filaments, but there are no thick filaments in this area. And there's a Z disc that passes through the center of each I band. There's a narrow H zone in the center of each A band that contains thick filaments, but no thin filaments. A mnemonic that will help you remember the composition of the I and H bands is that the letter I is thin, and so there's only going to be thin filaments in this area, whereas the letter H is thick, so there's only going to be thick filaments in this area. Supporting proteins that hold the thick filaments together at the center of the H zone form the M line. And this is named because it's in the middle of the sarcomeres. Now, myofibrils are built from three kinds of proteins. Contractile, which generate force during contraction. Regulatory proteins, these help to switch the contraction processes on and off. And structural proteins. Now these keep the thick and thin filaments in the proper alignment. They give the myofibril elasticity and extensibility, and they also link the myofibrils to the sarcolemma and the extracellular matrix. Okay, so the first two contractile proteins in the muscle, these are going to be myosin and actin. These, again, are going to be the thick and thin filaments. We're going to make up the thick and the thin filaments. Myosin is going to be the main component of the thick filaments. We can see one down here made up of the myosin molecule. And really, myosin functions as a motor protein in all three types of muscle tissue. So motor proteins will pull various cellular structures to achieve movement. And they do this by converting chemical energy in ATP to mechanical energy of motion. So we're going to produce force. So that's an important thing. So myosin converts chemical energy and it converts it into mechanical energy. Each myosin molecule is shaped like two golf clubs kind of twisted together. You can kind of see that here with this. So we can see the myosin tail is like a twisted golf club handle, and the tail is going to point towards the M line in the center of the sarcomere. And then two projections of each myosin molecule are called the myosin heads, kind of like the golf club ends. Then we have the thin filaments over here. These are going to be anchored to the Z discs, remember, and their main component is the protein actin. Individual actin molecules, as we can see here, these little yellow balls, these are going to be joined together to form an actin filament that is twisted into a helix. On each actin molecule is a myosin binding site. It's this little black dot on the yellow actin balls here, and this is going to be the part where a myosin head can attach. So these parts are going to attach to these little black dots, which are the myosin binding sites on actin. So those are our contractile proteins. Now there are smaller amounts of two regulatory proteins, troponin and tropomyosin, and these are also going to be parts on the thin filaments. So we can see them in this picture here. In relaxed muscle, myosin is blocked from binding to actin because tropomyosin covers those myosin binding sites on actin. So tropomyosin is going to be like this long orange strand that we can see here, and it's blocking those myosin binding sites on actin. So this is going to be in relaxed muscle. The tropomyosin strands are held in place by troponin molecules, these little blue globs. So tropomyosin is held in place by troponin. When calcium binds to troponin, it undergoes a change in shape, which moves the tropomyosin away from the binding sites on actin, and then muscle contraction will subsequently begin as myosin can now bind to actin. Okay? Now there are about a dozen of these structural proteins in muscle tissue, and these are going to contribute to the alignment, to stability, to elasticity, and the extensibility of the myofibrils. Extensibility. So we're just going to talk about a few key structural proteins today. First is titin. This is the third most plentiful protein in skeletal muscle. And each titin molecule connects the Z disc to the M line of the sarcomere. So it helps to stabilize the position of the thick filament. So stabilize the thick filament is what this one does, by connecting the Z disc to the M line of the sarcomere. Now the dense material of the Z discs contains molecules of alpha-actinin, and this binds the actin molecules of the thin filaments to titin. Molecules of myomesin, these are going to form the M line. Nebulin. This is a long, non-elastic protein that wraps around each thin filament and helps to anchor the thin filaments to the Z discs, and really helps to regulate the length of thin filaments during development. But really we're just going to say that this anchors thin filaments to the Z discs. And then we have dystrophin, which is probably the most important structural protein in my opinion. And this is going to be the one that you're going to want to remember here. So dystrophin links the thin filaments of the sarcomere to integral membrane proteins of the sarcolemma, which are attached in turn to proteins in the connective tissue extracellular matrix that surrounds the muscle fibers. So really, what, in like layman's terms or in simpler terms, what dystrophin does is that it links the sarcomere to the sarcolemma. So that the sarcomere, which is inside the entire muscle cell, so we'll say this is the entire muscle, and this is the sarcomere inside. When the sarcomere contracts, it contracts the entire muscle because it's going to be attached to the sarcomere, or sorry, to the sarcolemma. So that when the sarcomere contracts, it also contracts the entire muscle because it's going to be attached to the sarcolemma. If we didn't have dystrophin, something like this would happen. So we're going to get rid of these little blue dystrophin so that this is our general size of the muscle cell and the sarcolemma, but then we're going to have this contraction of the sarcomere inside, but that the muscle itself stays the same length because this is just going to contract inward and it's not attached to anything. So we need to have this dystrophin attaching the sarcomere to the sarcolemma so that the entire muscle can contract as a unit. So the sliding filament mechanism, this is a model that describes the process of muscle contraction at the molecular level. Now, muscle contraction occurs because the myosin heads will attach to and walk along the thin filaments at both ends of the sarcomere, progressively pulling the thin filaments towards the midline of the sarcomere. Now, as a result, the thin filaments will slide inward and meet at the center of the sarcomere. Structural proteins, these will pretty much hold everything together, and it will also allow the contraction to propagate through the entire muscle. So the thin filaments here in like the gold color, they're going to slide towards each other during contraction, and they can even move so far inward that their ends overlap too. Now, as these thin filaments slide inward, the I band and the H zone will narrow and eventually disappear completely, as you can kind of see in this maximally contracted muscle. However, the width of the A band and the individual lengths of the thick and thin filaments is going to remain unchanged. So as you can see here, we have the A band going from there to there. When we're partially contracted, A band goes from here to here, and fully contracted, A band goes from here to here. So the A band is never going to change length, and the length of the thick filaments and thin filaments themselves does not change length. But the H zone will shorten as contraction occurs, and the I band will also shorten as contraction occurs. Now, since these thin filaments are attached to the Z discs, when the thin filaments slide inward, the Z discs will come closer together and the sarcomere itself shortens. So shortening of the sarcomeres causes shortening of the whole muscle fiber, and in turn leads to shortening of the entire muscle. So here we have a contraction cycle, the sliding mechanism here. So at the onset of contraction, the sarcoplasmic reticulum releases calcium ions into the sarcoplasm, and they will bind to troponin. Troponin will move tropomyosin away from the myosin binding sites on the actin. And then once the binding sites are free, the contraction cycle, the repeated sequence of events that causes the filaments to slide, is actually going to begin. So here again in this picture, we could see the key that has calcium, which are the little purple circles. And let's go through this 4-step contraction cycle. So here at the top, we have step 1. This is ATP hydrolysis step. This is where the myosin head, which includes an ATP binding site, as we could see here, and there's also an ATPase on these myosin heads. Remember, ATPase, this is going to be an enzyme that hydrolyzes ATP into ADP and a phosphate group, as we could see here. ATP into ADP plus a phosphate group. So this kind of reaction, this is hydrolysis reaction, is going to reorient the myosin heads, and it's also going to energize the myosin head. So they're going to hydrolyze that ATP, hydrolyze it to ADP and phosphate, and kind of get them in position. So as you can see here, they're kind of in perfect position here to bind. Step 2 over here, this is going to be the attachment of the myosin heads to actin, and it forms what we call cross bridges. So the energized myosin head, as we can see here, it still has this ADP attached, but it's going to be energized, and it's going to attach to these myosin binding sites on the actin. And it also releases this phosphate group at the same time. Now, when the myosin head attaches to actin during contraction, this is referred to as a cross bridge. So one thing I forgot to mention is that this contraction cycle can't begin until calcium binds to that to troponin, and it moves the tropomyosin. Because we can't have a contraction cycle if the actin is completely covered. Contraction cycle can only begin once calcium is released around the sarcomeres, and the calcium binds to troponin in order to move tropomyosin away from those myosin binding sites on actin. Okay, moving on to step 3 now here. Step 3 is called the power stroke step. So after these cross bridges form, this is when the power stroke occurs. So during the power stroke, the site on the cross bridge where ADP is bound is going to open. So as a result, the cross bridge rotates and releases ADP. So you can kind of see here that the myosin heads will kind of turn their heads in. kind of rotate and they release the ADP, sorry. Now this cross bridge is going to generate force as it rotates towards the center of the sarcomere, sliding the thin filament past the thick filament toward the M line. So the thin filament is going to be moving this way. And that brings us to step 4 here on the left, and this is going to be the detachment of myosin from actin. So at the end of the power stroke, the cross bridge remains firmly attached to actin until it binds to another molecule of ATP. Now, as ATP binds to the ATP binding site on the myosin head is going to detach from actin. Now, this contraction cycle is going to continue and it's going to repeat as myosin ATPase hydrolyzes the newly bound molecule of ATP, and it's going to continue as long as ATP is available and calcium levels near the thin filament is sufficiently high. These cross bridges will keep rotating back and forth with each power stroke, pulling the thin filaments towards the M line. So as we know, an increase in calcium concentration in the sarcoplasm starts muscle contraction, and then a decrease is going to stop muscle contraction. When a muscle fiber relaxes or is relaxed, the concentration of calcium in the sarcoplasm is going to be very low. However, there's still a large amount of calcium that's stored within these sarcoplasmic reticulums. So we still have a lot of calcium stored within there. So as a muscle action potential propagates or moves along the sarcolemma and then down into the T-tubules, it causes these calcium release channels to open, and it allows calcium to be completely released into the sarcoplasm. So when these channels open, calcium is going to flow out of the sarcoplasmic reticulum into the sarcoplasm around the thick and thin filaments. So we can see the calcium moving out into the sarcoplasm and then all around the thick and thin filaments here. So as a result, the calcium concentration in the sarcoplasm rises like tenfold or more. Now those released calcium ions are going to combine with troponin. Remember this blue glob here, so here we can see calcium ions bind to troponin, causing it to change shape. So we get that conformational change of troponin, and it moves tropomyosin away from the myosin binding sites on actin. Now, once these binding sites are free, the myosin binding or the myosin heads will bind to them to form the cross bridge. And then this is when that contraction cycle begins, that contraction cycle we just talked about. Now, these events leading up to the contraction cycle are referred collectively as the excitation-contraction coupling, as these are going to be the steps that connect excitation, or the action potential propagating along the sarcolemma and into the T-tubule, to contraction, the sliding of the filaments. So we have excitation, meaning the muscle action potential moving along the sarcolemma, and also down into the T-tubules. And then the contraction part is the sliding filament mechanism. This figure here shows the length-tension relationship of skeletal muscle, which indicates how the forcefulness of muscle contraction depends on the length of sarcomeres within a muscle before contraction begins. So here we have the resting sarcomere length. So this is going to be the length of sarcomeres within a muscle before contraction begins. And here we have the developed tension on the y-axis. So with a sarcomere length of about 2 to 2.4, it's kind of like here to here approximately, or 2.2. This is going to be very close to the resting length in most muscles. This is where the zone of overlap in the sarcomere is optimal. And this is where the muscle fiber can develop the maximum tension here. Now notice that the maximum tension level, the 100% here, this occurs when the zone of overlap between the thick and thin filaments extends from the H zone to one end of a thick filament. Here we have that zone of overlap. Now outside of that optimal overlap, either more overlap like here or less overlap like here, this causes the possible tension to be quite considerably lowered. Now the neurons that stimulate skeletal muscle fibers to contract are called somatic motor neurons. Now each somatic motor neuron has a thread-like axon that extends from the brain or spinal cord to a group of skeletal muscle fibers. Now muscle action potentials arise at the neuromuscular junction, also known as the NMJ. This is going to be the synapse between the somatic motor neuron and skeletal muscle fiber. So as an action potential comes down the neuron, comes to this neuromuscular junction, it brings about an action potential in the muscle also. There are voltage-gated calcium channels that will open, resulting in this massive influx of calcium. This is going to be at the end of the neuron axon. Neurotransmitters will then be released into the synapse and bind to ligand-gated sodium channels at the motor end plate, which is the muscle side of the neuromuscular junction. Now, without this series of events, muscle contraction would not even be possible at all. So let's kind of go over what this synapse looks like, what the neuromuscular junction looks like. Now first, let's go over a synapse. So the term synapse is a region where communication occurs between two neurons or between a neuron and a target cell. In this case, our target cell is going to be a muscle. At most synapses, there's going to be a small gap, which is called a synaptic cleft, that separates the two cells. So they're not actually touching. There's a small space between them. Now, because the cells don't physically touch, the action potential can't just jump or just gap between from one cell to another. Instead, the first cell communicates with the second cell by means of a chemical messenger, which is called a neurotransmitter. So the first cell essentially has to talk to the second cell by these chemicals called neurotransmitters. At the neuromuscular junction, which we can see here, we can see the axon from a somatic motor neuron, And then here we have our muscle fiber. And here we have a larger picture of a neuromuscular junction, kind of just zooming in to this region of communication between these two cells. So at the neuromuscular junction, the end of the motor neuron, which is called the axon terminal, divides into a cluster of synaptic end bulbs. And within each of these synaptic end bulbs, there's going to be hundreds of these membrane-enclosed sacs called synaptic vesicles. And these synaptic vesicles are going to be filled with neurotransmitters, those chemical messengers. Now, there usually are several different types of neurotransmitters used at different synapses, but between a neuron and a muscle cell, acetylcholine is used, abbreviated ACH. So this is going to be our chemical messenger at the neuromuscular junction. Now the region of sarcolemma just opposite the synaptic end bulbs is called the motor end plate. So this is part of the muscle. This is the muscle fiber part of the neuromuscular junction. Now within each motor end plate, we can see in this bottom picture here, there's going to be like 30 to 40 million of these acetylcholine receptors. So they're going to be these little green receptors here. These are going to be acetylcholine receptors. This is going to be an integral membrane protein, and this is where acetylcholine is going to very specifically bind. So here we can see the events at the neuromuscular junction. Here we have this purple arrow is the nerve impulse. So we have a nerve action potential coming down the axon to the synaptic end bulb here. And once that action potential comes to the end, these voltage-gated calcium channels open in the nerve, allowing calcium into the synaptic end bulb. Once calcium enters the synaptic end bulb, it exocytoses these synaptic vesicles that are filled with acetylcholine. So all this acetylcholine is released into this space here, which is the synaptic cleft, the space between the neuron and the muscle. All this acetylcholine is released into the synaptic cleft, and acetylcholine is going to cross the cleft and then bind to these acetylcholine receptors on the muscle side, the motor end plate. Acetylcholine will bind to the receptors, which causes these receptors to open and allow sodium into the motor end plate of the muscle. And this is how a muscle action potential is produced. Now this is what a neuromuscular junction looks like from an electron micrograph. Here you can see the neuron. We can also see the muscle fibers. And here we can also see that neuromuscular junction here with the synaptic end bulbs attaching to the skeletal muscle fibers at their motor end plates. So a nerve impulse, also known as a nerve action potential, elicits a muscle action potential in these following steps. So we're kind of putting everything together now that we've kind of learned about in the past few slides. So we're going to start with step 1 up here. This is going to be the release of acetylcholine. So the arrival of the action potential nerve impulse at the synaptic end bulbs stimulates these voltage-gated channels to open in the synaptic end bulb. Now, because calcium ions are in a much higher concentration in the extracellular fluid, the calcium ions are going to flow inward through the open channels into the synaptic end bulb. So we have calcium going in here. Now, calcium ions stimulate these synaptic vesicles within the end bulb. So here we have these labeled synaptic vesicles are filled with acetylcholine. Calcium is going to cause these synaptic vesicles, which contain acetylcholine, to undergo exocytosis. So we can see this synaptic end bulb here, or sorry, this synaptic vesicle is releasing its acetylcholine into the synaptic cleft area. Now these acetylcholine molecules will cross the synaptic cleft. So they cross this whole cleft here. And they cross the cleft and they reach the motor end plate. So essentially that's all that's happening with step 1 here. Nerve impulse arrives at the axon terminal, triggers the release of acetylcholine. Step number 2 is the activation of the acetylcholine receptors on the motor end plate. So the binding of 2 molecules of acetylcholine to the receptor on the motor end plate opens ion channels that allow sodium to cross the membrane of the muscle. Step 3 here is the production of the muscle action potential. So the inflow of sodium down its concentration gradient, down its electrochemical gradient, makes the inside of the muscle more positively charged, and the change in the membrane potential is going to trigger a muscle action potential. Now the muscle action potential is going to propagate along the sarcolemma and then down into these systems of T-tubules. Now this causes the sarcoplasmic reticulum to release its calcium into the sarcoplasm, and the muscle fiber contracts by that sliding filament model. So here we have calcium binding to troponin. We have that sliding filament model contraction. We kind of just talked about this all very quickly before. And then once it's finished, calcium active transport pumps are actually going to pump calcium back into the sarcoplasmic reticulum to relax muscle in the end. Now acetylcholine here within this synapse, this effect of acetylcholine is going to last only very briefly, and this is because acetylcholine is kind of quickly broken down by an enzyme called acetylcholine esterase. This is also often abbreviated ACHE. So this is the enzyme that breaks down acetylcholine in the synaptic cleft after it's bonded to the acetylcholine receptor and it's kind of done its job. Then acetylcholine is broken down. If another nerve impulse reaches the neuromuscular junction, then more acetylcholine is going to be released from the synaptic, from the end bulb in the nerve, and then this whole cycle is going to repeat again. Now, neuromuscular junctions are usually found near the midpoint of a skeletal muscle fiber. So the muscle action potentials that arise at the neuromuscular junction can propagate or move towards both ends of the fiber. So this is going to allow almost like nearly simultaneous stimulation or simultaneous activation, and so the entire muscle fiber can contract all at once. Unlike most cells of the body, skeletal muscle fibers will often switch between low levels activity, like when they're relaxed and only using a modest amount of ATP, and switch to very high levels activity, so when they're contracting, using ATP at a very rapid pace. Now, a huge amount of ATP is needed for the contraction cycle to pump calcium into the sarcoplasmic reticulum and for other metabolic reactions involved in muscle contraction. Now, muscle fibers have three ways to make ATP. This is by creatine phosphate, by anaerobic glycolysis, and by cellular respiration. Now, the use of creatine phosphate is actually really unique to muscle fibers to make ATP. So the use of creatine phosphate specifically for ATP production is very unique to muscle fibers, but all body cells can make ATP by the reaction of anaerobic glycolysis and the aerobic cellular respiration. Now, when muscles are relaxed, they produce more ATP than they need for resting metabolism. Now, most of the excess ATP, so we can kind of see here, is used to synthesize creatine phosphate. Creatine phosphate is a really energy-rich molecule that's found in muscle fibers. Now, the enzyme creatine kinase, this is also sometimes abbreviated CK. This is an E here. Creatine kinase. This is an enzyme that catalyzes the transfer of one of the high energy phosphate groups from ATP to creatine. And this forms ADP and a high energy creatine phosphate. So again, this is going to be in relaxed muscle. When muscle is relaxed, you're going to be producing a lot more ATP than you need. So creatine here, this is a really small amino acid-like molecule that's synthesized in the liver, kidney, and pancreas, and then transported to muscle fibers. Now, when muscle activity continues or resumes if you're resting, the supply of creatine phosphate within the muscle fiber is going to be depleted because creatine phosphate will then be used to extract that ATP from, and energy is used for muscle contractions. But once all of that high energy creatine phosphate is used and depleted, then glucose is catabolized to generate ATP. So ATP from creatine phosphate is going to be a very quick duration of energy. It's very fast to provide ATP, but it also, that energy doesn't last very long. So it only lasts about 15 seconds, the energy provided from creatine phosphate. So once that's all used up, then we're going to use glucose. So glucose passes quite easily from the blood into contracting muscle fibers, and this is by facilitated diffusion. And we also get glucose from the breakdown of glycogen within muscle fibers. So kind of two different pathways to get to glucose here. Now, the entire process by which the breakdown of glucose gives rise to lactic acid when oxygen is absent or at low concentrations, this is referred to as anaerobic glycolysis. So anaerobic, this term here means little to no oxygen. So in very low oxygen conditions, your glucose is going to be converted into lactic acid. whereas aerobic here it means oxygen requiring. Now compared to aerobic respiration, which we'll go over right shortly, this anaerobic glycolysis produces fewer ATPs. We're only going to get about 2 ATP per glucose molecule. But this process is a lot faster than aerobic respiration. And it can also occur when oxygen levels are low. So anaerobic glycolysis provides enough energy for about 2 minutes. And this is at maximal muscle activity. Now, if sufficient oxygen is present, though, pyruvic acid is going to enter into the mitochondria and is actually going to take a different route than lactic acid. So it's only going to go this way when there's no oxygen. If there is oxygen present, then we're going to come over here. So oxygen present. We're going to come over here to aerobic respiration. So in the mitochondria, aerobic respiration occurs, and aerobic respiration is the series of oxygen requiring reactions. So this is going to be the Krebs cycle. I'll just put this down here, but we will go over this a little bit later, especially when we go through metabolism. But the Krebs cycle and the electron transport chain. Sorry, the electron transport chain. These are going to occur in the mitochondria, and these are going to produce ATP, quite a lot of ATP, 30 to 32 ATP per glucose. We're also going to produce carbon dioxide, water, and heat. Therefore, when oxygen is present, glycolysis followed by the Krebs cycle and the electron transport chain will occur. The inability of muscle to maintain force of contraction after prolonged activity is called muscle fatigue. Now this is could be due to a lot of things. First, it could be due to an inadequate release of calcium ions from the sarcoplasmic reticulum. Could be due to the depletion of creatine phosphate, of oxygen and nutrients. Could be due to a buildup of lactic acid and even a buildup of ADP. Or it could also be due to an insufficient release of acetylcholine at the neuromuscular junction. So during prolonged periods of muscle contraction, increases in breathing rate and blood flow enhance oxygen delivery to muscle tissue. The term oxygen debt has been used before to refer to the added oxygen over and above the resting oxygen consumption that's taken into the body after exercise. So this extra oxygen is used essentially to pay back or restore metabolic conditions to the resting level, and it does this in 3 ways. First, to resynthesize creatine phosphate and ATP in muscle fibers. Otherwise, we can think of it as replenishing that creatine phosphate stores. To convert lactic acid back into glycogen stores in the liver. And also to replace the oxygen removed from the myoglobin. Reload it back onto the myoglobin. Now really a better term for oxygen debt would be recovery oxygen uptake. And this is because the elevated oxygen consumption after exercise is to bring the body back to normal. Even though each skeletal muscle fiber has only a single neuromuscular junction, the axon of a motor neuron branches out and forms neuromuscular junctions with many different muscle fibers. So a motor unit consists of the somatic motor neuron plus all of the skeletal muscle fibers that it stimulates. A single somatic motor neuron makes contact with an average of about 150 different skeletal muscle fibers. So this is per somatic motor neuron. And so this is that so that all of the muscle fibers in one motor unit are going to contract all in unison. So let's talk about twitch contractions. So a twitch contraction is the brief contraction of all muscle fibers in a motor unit, and this is in response to a single action potential in its motor neuron. So this little diagram on the right is a record of a muscle contraction, and we call this figure a myogram. And twitches from skeletal muscles can last anywhere from 20 milliseconds even up to 200 milliseconds. So this little arrow here is indicative of the application of stimulus. This little arrow here. So note that there is a brief delay between the activation of the stimulus here, time 0 on the graph, and the beginning of a contraction. So this is kind of called the latent period. So during this latent period, the muscle action potential sweeps over the sarcolemma, and calcium ions are released from the sarcoplasmic reticulum. That's what happens in this latent period. Muscle action potential sweeps over the sarcolemma, and the calcium ions released from the sarcoplasmic reticulum. Then that brings us to the second phase, which is the contraction period. This lasts usually around 10, and it can be as long as even 100 milliseconds. Now during this time, this is when calcium binds to troponin, the myosin binding sites on actin are being exposed, and the cross bridges are forming. It's during this period where we get peak tension develop in the muscle fiber. Now during the third phase, which is the relaxation period, this also lasts about 10 milliseconds to as long as 100 milliseconds. It's during this period that calcium is being actively transported back into the sarcoplasmic reticulum. The myosin binding sites are being covered by tropomyosin. The myosin heads detach from actin. And here we can see the tension in the muscle is decreasing. Now, if two stimuli are applied, one immediately after another, the muscle will respond to the first one, but it can't respond to the second one. When a muscle fiber receives enough stimulation to contract, it temporarily loses its excitability, and it cannot respond for a period of time. So this period of lost excitability is called the refractory period. Now, this period of lost excitability, this refractory period, is characteristic of all muscle and nerve cells. The duration of this refractory period really varies with the type of muscle involved, so it's not the same everywhere. For example, skeletal muscle has a relatively short refractory period of just about 5 milliseconds, whereas cardiac muscle has a much longer refractory period of about 300 milliseconds. Not that you have to memorize those numbers or anything, but just varies between different muscle types. When a second stimulation occurs after the refractory period of the first stimulus is over, but before the skeletal muscle fiber is relaxed, the second contraction will actually be stronger than the first one. Now, when this happens, and where stimuli arriving at different times causes larger contractions, this is called wave summation. The result of being stimulated at about 20 to 13 times per second in a skeletal muscle causes this sustained but wavering contraction called unfused tetanus. So this is when we get stimulation of like 20 to 30 stimulations per second in skeletal muscle. You get kind of like a sustained contraction. But it kind of wavers, so it's not complete contraction all the time. It's a wavering contraction. Now, when a skeletal muscle fiber is stimulated at a higher rate, like 80 to 100 times per second, it does not relax at all. So this is the result, or this results in what's called fused tetanus. This is a complete sustained contraction in which the individual twitches can't be detected. There is no relaxation. Whereas in unfused tetanus, there's going to be slight little deviations, slight relaxations between these contractions. So this figure shows a single twitch here on the left. And you can see there's a distinct contraction period and a distinct relaxation period. The next one here is wave summation. So this is where we have that second action potential, or a second stimulation occurs quite quickly after the first one. And you can see that this second contraction occurs slightly after the first one, and it causes a larger force of contraction. Unfused tetanus, this produces this jagged curve. You can see here we have slight relaxations between contractions, or partial relaxations, we could say. And these are going to be partial relaxations between stimuli. Then we get fused tetanus, and again, this is where we get like 80 to 100 stimuli per second. We can see this myogram line is going to be very steady and sustained. No partial relaxation. Now, the process in which the number of active motor units increases is called motor unit recruitment. Now, typically, the different motor units of an entire muscle are not stimulated to contract in unison. So while some motor units are contracting, others are relaxing. So your entire muscle is not all contracting at once. So this is a pattern of motor unit activity that delays muscle fatigue and allows contraction of a whole muscle to be sustained for longer periods. In this way, the weakest motor units are going to be recruited first, with progressively stronger motor units added if the task requires more force. Even at rest, skeletal muscle exhibits what's called muscle tone. This is going to be a small amount of tension or tightness in muscle, and these are due to really weak involuntary contractions of motor units. Muscle tone is established by neurons in the brain and spinal cord that excite the muscle's motor neurons. Now, when the motor neurons serving a skeletal muscle are damaged or cut, the muscle becomes flaccid. So this is going to be a state of limpness in which all muscle tone is lost. Now, muscle contraction may either be isotonic or isometric. In isotonic contraction, the tension, which is the force of contraction, developed in the muscle remains almost constant while the muscle changes length. This type of contraction is used for body movements and for moving objects. There's going to be two types of isotonic contraction. These are concentric and eccentric. Then the other type of muscle contraction is isometric contraction. In an isometric contraction, the tension generated is not enough to exceed the resistance of the object to be moved, and so the muscle does not change length. So if the tension generated in concentric isotonic contraction is great enough to overcome the resistance of the object to be moved or overcome the weight of the object, then the muscle shortens and pulls on another structure, such as a tendon, to produce movement. And this will reduce the angle at the joint. So like picking up a book. So picking up a book is going to involve concentric isotonic contractions of the biceps brachii muscle in the arm. Now when the length of the muscle increases, this is during a contraction, then the contraction is going to be considered eccentric isotonic contraction. Now during an eccentric contraction, the tension that's exerted by the myosin cross bridges resists the movement of a load and is going to slow the lengthening process. So it's like lowering a book without just completely dropping it quickly. And then we have the isometric contraction. In this type of contraction, the tension generated does not exceed the resistance of the object to be moved, so it doesn't exceed the weight of the object, and so the muscle isn't going to change its length. Holding an object steady uses this type of contraction. Different skeletal muscle fibers contract and relax at different speeds, and they also vary in which metabolic reactions they use to generate ATP and how quickly they fatigue. For example, a fiber is going to be categorized as either slow or fast, depending on how rapidly the ATPase in its myosin heads hydrolyzes ATP. Now, based on these structural and functional characteristics, skeletal muscle fibers are classified into three main types. So these three main types of muscle fibers are slow oxidative fibers, fast oxidative glycolytic fibers, and fast glycolytic fibers. So first off, we'll talk about the slow oxidative fibers. Now these are going to appear dark red, and this is because they contain large amounts of myoglobin and many capillaries. They have many large mitochondria as well. And they mainly generate ATP by aerobic respiration. The ATPase in the myosin heads will hydrolyze ATP relatively slowly, and the contraction cycle proceeds at a slower pace, than the fast fibers. So it kind of explains why they're called slow oxidative fibers, because they use oxidative respiration or aerobic respiration. So aerobic respiration is an oxidative type of metabolism, and the ATPase in the myosin heads hydrolyzes ATP relatively slowly. So that's where they kind of get that slow from. So you can see here on this transverse section picture, you can see these slow oxidative fibers appear very dark. So they're going to be this very dark red color. Okay, so let's erase that. And we'll move on to the fast oxidative glycolytic. So the fast oxidative glycolytic are typically the largest fibers. And they also contain large amounts of myoglobin and several capillaries, though not as many as the slow oxidative fibers. So we're going to have large amounts of myoglobin, several mitochondria, or several capillaries, sorry. And these fibers also appear red or pink in color, so kind of a lighter red than the slow oxidative fibers. And they can generate, well, actually we'll put largest, and we'll also put pink here. They do generate a considerable amount of ATP by aerobic respiration. And this gives them really a moderately high resistance to fatigue. So slow oxidative fibers are also going to be slow to fatigue. Now, because their intracellular glycogen level, back to the fast oxidative glycolytic, they have an intracellular glycogen level that's very high. They can also generate ATP by anaerobic glycolysis. So they kind of do both. Now they're considered fast because the ATPase in their myosin heads hydrolyzes ATP about 3 to 5 times faster than in the slow oxidative fibers. So it's a bit of a quicker ATPase action. We'll just put then the SO, slow oxidative fibers. Okay, then we're going to finally discuss the fast glycolytic. I guess too, on the picture on the right, you can see, we'll finish erasing this, but you can see those fast oxidative glycolytic fibers, which are still a little pink, but they're a lot lighter than those slow oxidative fibers. So we can see those right here. They're a little bit lighter in color, but still come up a little bit pinky. Okay, lastly, onto the fast glycolytic fibers. These ones here. These ones have a low myoglobin content. Sorry, myoglobin. And they have relatively few blood capillaries. They have very few mitochondria. And They generally appear white in color. They contain large amounts of glycogen, so lots of glycogen. And they generate ATP mainly by glycolysis. So they can generate ATP rather quickly this way. Now, because they can generate ATP very rapidly, these fast glycolytic fibers will contract strongly and quickly, but they also fatigue quickly. So we can see these in the picture on the right. These fast glycolytic fibers appear very white, especially compared to these slow oxidative fibers and also the fast oxidative glycolytic fibers. So you can tell with these slow oxidative fibers and then a little bit with the fast oxidative glycolytic fibers, they require a lot of blood vessels to bring a lot of oxygen to these muscle fibers in order to use these aerobic respiration means of creating ATP. Whereas the fast glycolytic fiber doesn't require these blood vessels to bring oxygen because their metabolism methods just have to do with glycolysis, which is an anaerobic type of way to create ATP. An interesting thing about skeletal muscle is that stretching cold muscles does not increase flexibility and actually might cause injury. Tissues stretch best when slow, gentle force is applied at elevated tissue temperatures. So external heat may be used, but 10 or more minutes of muscular contraction is really a good way to raise muscle temperature prior to stretching. Strength training refers to the process of exercising with progressively heavier resistance for the purpose of strengthening the musculoskeletal system. Now, this activity results not only in stronger muscles, but many other health benefits as well. Very briefly on cardiac muscle. So the principal tissue in the heart wall is cardiac muscle tissue. Between the layers of the cardiac muscle fibers, the contractile cells of the heart are sheets of connective tissue that contain blood vessels and the conductions of the heart. Cardiac muscle tissue has the same arrangements of actin and myosin and the same bands that we've seen in skeletal muscle tissue, the same zones and Z discs that we've seen before. So we can see these striations still here in the cardiac muscle. However, there's also this other thing that's called intercalated discs. These are going to be very unique to cardiac muscle fibers. Now, these are going to be really microscopic structures that are irregular transverse thickenings of the sarcolemma, and these connect the ends of cardiac muscle fibers to one another. So you can see this right here, these connections between adjacent cardiac muscle fibers. Now, these discs contain desmosomes, which hold these fibers together, and they also contain gap junctions. And these gap junctions allow muscle action potentials to spread from one cardiac muscle fiber to another. Cardiac muscle tissue has endomysium and paramysium, but it does lack epimysium. Now, like cardiac muscle tissue, smooth muscle tissue is usually activated involuntarily. And there are actually two types of smooth muscle tissue. And now of those two types, the more common type is called the visceral smooth muscle tissue. Now this is a single unit it's a single unit type of smooth muscle, so we can see this one right here, just with a single cell here. This type of smooth muscle is found in the skin and in tubular arrangements that form parts of the walls of small arteries and veins and hollow organs like the stomach, intestines, uterus, and urinary bladder. Now the second type of smooth muscle tissue is the multi-unit smooth muscle. Actually, I'll change the color here. Now this consists of individual fibers, each with its own motor neuron terminals and with a few gap junctions between neighboring fibers. So we can see this actually here with having their each individual neuron attached to them. Now, stimulation of one of the visceral smooth muscle tissue cells causes contraction of many adjacent fibers, but stimulation of one multi-unit fiber causes contraction of that fiber only. Now, the sarcoplasm of smooth muscle fibers contains both thick and thin filaments, but they're not arranged in orderly sarcomeres as we've seen in striated muscle. Smooth muscle fiber also contain what are called intermediate filaments. Now, because these various filaments, these thick and thin filaments, don't have a regular pattern of overlap, smooth muscle fibers don't exhibit striations. The thin filaments will actually attach to structures called dense bodies. This is where thin filaments attach. And these dense bodies are going to be functionally similar to Z discs that we've seen in the striated muscle fibers. Some dense bodies are dispersed throughout the sarcoplasm, whereas others are going to be attached to the sarcolemma. Once a muscle cell has differentiated into a mature muscle cell, it cannot undergo mitosis. Now, because of this, muscle tissue growth after birth mainly occurs by hypertrophy. So this is going to be the enlargement of existing cells. And it will go through hypertrophy rather than hyperplasia, which is an increase in the number of fibers. Or in the number of cells. In other tissues, hyperplasia is the increase in the number of cells by mitosis, by the division of existing cells. And because these mature skeletal muscle fibers can't undergo mitosis, hyperplasia generally doesn't occur. So after birth, we're mainly going to just be growing the existing cells. Sorry, not supposed to say after birth there. Here, I'll quickly rewrite it so it's a little bit more legible. Now, remember that muscle cells do have satellite cells, which are muscle stem cells. So satellite cells do divide slowly, and they can fuse with existing fibers to assist both in muscle growth and the repair of damaged fibers. So In this way, skeletal muscle tissue can regenerate, but really just to a limited extent. Cardiac muscle tissue also can regenerate less than skeletal muscle, but again, cardiac muscle regeneration is extremely limited. Smooth muscle tissue, just like skeletal and cardiac muscle tissue, are going to undergo hypertrophy. And new smooth muscle fibers can arise from cells called pericytes. And these are going to be stem cells found in association with blood capillaries and small veins. Now, just like the other tissues and systems of the body, aging has an effect on muscle tissue. So between the ages of about 30 to 50, there's about 10% of our muscle tissue that's replaced by fibrous connective tissue and adipose tissue or fatty tissue. Between 50 to 80 years of age, another 40% of our muscle tissue is replaced. Now this has several consequences, including strength and flexibility decrease, a decrease or a slowing of reflexes, and also slow oxidative fiber numbers are going to increase. They're going to replace those fast glycolytic fibers.